My name is Tiffany Wolfel, and I'm a current dual master's student in public health and social work at the University of Washington. And I have the opportunity to be here tonight because of the special collaboration that Town Hall has with the UW's Engage program. Together they host the series that's called UW Science Now, which is part of the Seattle Science Lectures. Sponsors include Microsoft and KPLU. Each speaker programs will run about 25 minutes, including 5 minutes for your questions. We ask that you do come to the mics at the end of each of the rows up here so that we can make sure to record any of your questions or comments. Before I introduce our first guest, I'd like to mention some other events with our science series. First of all, join us tonight upstairs at 7:30 for Janet Sadiq Khan and Mayor Ed Murray, who will discuss implementing an urban revolution, and that'll be in the Great Hall. Also at 7:30 downstairs here will be Sean Carroll giving a talk called Understanding Life's Ecological Roles. There will also be 2 talks for the Engage series next Monday at 6 p.m. and I'll be one of the speakers. My talk is on social media and the ethical use of research within social media. And following me will be Marshall Styczynski, and he will be discussing how to make physics more accessible. So now for our first guest, oceanography and astrobiology graduate student Max Showalter looks at how bacterial life in sea ice adapts to the extreme conditions of the Arctic and how we might use our knowledge of these organisms to improve our search for life elsewhere in our universe. Please give a warm welcome to Max Showalter.
Great, thanks Tiffany. So like she said, my name is Max and I'm going to be talking to you for the next few hours about finding life when the trail goes cold. Okay, so I'm going to start out by taking us on a journey. So this is the solar system here. You are here on Earth, and we're going to be talking about places out here near the planet Jupiter. And specifically, we're talking about Europa. So Europa is a moon of Jupiter, and it's a very special moon, and it's been in current events recently. So we're going to do a little quiz that shouldn't be too hard, hopefully. And the question is, how much money did Congress recently earmark for NASA's Europa Clipper project? So in the 2015 fiscal year budget, they approved this really awesome budget for NASA, which included money for this Europa Clipper mission to go to Europa. So I have 3 options here. So how many people think it was $1.75 million? Okay, a few people. How about $17.5 million? A few more? Okay, $175 million? Yeah, so the majority rules, you're right, it's $175 million. So you might be asking, why are we spending so much money to go to Europa? What's so special about the moon Europa? So Europa is really special because it has a global ocean. So you can see here in this GIF a comparison about the amount of water on Earth as compared to Europa. So Earth is on the right, Europa on the left, and those little blue balls up there are representing how much liquid water is on the planet. And that's really significant when we're looking for life in our solar system and in outer space in general, because everywhere that we've found water on Earth, we've found life. Usually in the microbial form, so bacteria, germs, but everywhere that we've found water on Earth, we've found life. So that makes it seem easy, right? We just go to Europa, there's a high chance that we might find life there, but there's another problem. In addition to it being 390 million miles away from the sun, it's covered in this huge icy crust. So since it's so far from the sun, it's really cold on Europa, and it has this crust of frozen ocean on top of it, kind of like our own Arctic Ocean, for example, except it's 8 kilometers thick. So as a comparison, the Antarctic ice cap is about 4 kilometers thick. So this is about twice as thick. So the question is, when we get to Europa, how do we get through that ice, or can we find a sample of life in that ice? So there's an even bigger problem, just when you thought we had so many problems with getting to Europa and finding life. It's actually really hard for us to identify life even here on Earth. So when we're looking in outer space for life or on Earth for life, we're looking for what's called a biosignature. So a biosignature is any indicator of past or present life. This can take a lot of forms. For example, on Earth, we're looking for fossils of things like bacteria. We're looking for things like stromatolites, which are layers of microbial mats, so where lots of bacteria have been laid down on a surface and then fossilized over time. Gases that might represent life, for example, are a biosignature. But even on Earth, it's really hard for us to identify where life exists, in certain cases where life has existed in the past. So to exemplify this, I have this picture here, which is from a meteorite that landed in Antarctica several thousand years ago. And it was discovered around the 90s. Some of you might remember when this was really big in the news. It was significant because this meteorite is from Mars. And as you can see in this picture, and some people speculated, this kind of looks like a bacterium. So there are a lot of people who thought that this was definitive evidence of life coming from Mars in the form of this fossilized bacterium. So not shown in this image is a scale, which shows how really tiny this bacterium is, even smaller than a typical bacterium. And it's since been kind of rebuked as evidence for life, and it's kind of generally thought that this is just an artifact of the rock itself. So even when we have a sample in our hand, it's really hard for us to tell that there's anything that was alive or is living. So one challenge for you as you walk out of this talk this evening is to think about our definition of life. How would you define life? When is something alive? Is a virus alive? That's part of the problem when we're trying to figure out biosignatures. Fortunately, when we're talking about Europa, which has this salty ocean on it and an icy crust on top, we have a really good analog site on Earth, a site that's really similar. And it's the Arctic up here. So up here is Alaska, just to orient you, in northern Canada. Greenland's over here, and Russia up here, and we're looking at the North Pole right here. So you can see in the Arctic, we have this huge ice cap that some of this ice stays for multiple years. Most of it is just frozen ocean that refreezes every year and melts in the summer. So it's a lot like Europa in that we have this thick ice crust. Granted, it's not 8 kilometers thick, but usually about 2 to 3 meters thick at its thickest, and then a salty ocean underneath. And this is where I do most of my work. So you might think ice is an inhospitable environment, but actually there's lots of life that lives in ice. This is a picture of an ice core that was taken up in Barrow, Alaska. So this is sea ice, or frozen ocean, that's wintered through an Alaskan winter, and even in the spring here, we have this discoloration at the bottom of the ice. So this is where the seawater hits up at the bottom of the frozen ice on top. And you can see from this discoloration that there's things inside the ice, and that's actually algae. So photosynthesizing small single-celled organisms that live in the ocean, and they're photosynthesizing in that ice. So light is penetrating through the ice and supporting a lot of biomass, a big ecosystem here in the ice. So here's a close-up picture of that. So we're zooming in on that ice core now. These little green things in here are the algae. And on the outside here, this is solid ice. And the algae are actually living inside of liquid water pockets inside the ice. These are called brine pores. And they're really salty, which keeps them from freezing at low temperatures. So these organisms live inside these really salty liquid pockets in the ice, and that allows them to persist in that really extreme environment. So in addition to algae, which are a little bit bigger, we also have bacteria inside of the sea ice. So bacteria are a lot smaller. They're about 100 times thinner than a human hair in their longest length. And so this is another picture of ice that's been zoomed in. So for scale, this is about 100 microns, which is about the thickness of a human hair. And this is solid ice here, here, and here. And this is that briny liquid pocket that, like we saw in the last picture with the algae. If we zoom in on this red square, here's another briny liquid pocket, and especially right here, there's a little bit more volume of brine. And you can't really see it in this picture, but there's actually bacterium hiding inside of there. So again, this scale is 10 times smaller, so this is 10 microns, or about 1/10 the thickness of a human hair. And if we stain the bacterium that's living in here with a special dye that glows under a specific light, we can see this little blue dot here is a bacterium living inside of that Arctic sea ice. So you might imagine if we're looking for that little blue dot in some Arctic sea ice, it's hard, but now multiply that by looking for that little blue dot of bacterium in sea ice that's 8 kilometers thick and 390 million miles away. It's not an easy task to do. So how do we look for a needle in a haystack? How do we hunt for something that might be right in front of us when we get there, when we spend all this money to get there, but we can't necessarily see it? So I'll give you an example of when there's life right in front of us, but we can't see it until it moves. So movement might be a good means of identifying life in these extreme environments. If we can see something move, that's kind of like a waving crowd. It's an unambiguous biosignature for us. So this is where my project comes in. NASA loves outrageous acronyms. So this project has the acronym SHAMU. It's not about the whale, although we have a nice whale on our microscope. And SHAMU stands for Submersible Holographic Astrobiology Microscope with Ultra Resolution, which is a lot of nouns, but don't worry, I'm going to go through it and explain a little bit what that means. So this project is led by a group down at Caltech and the NASA Jet Propulsion Laboratory, and our team here at UW collaborates on sort of the microbiology and oceanography of it. So let's start out with what is a hologram. So the idea here is we have this microscope that uses holography to look for bacteria swimming in samples. So what is a hologram and why is it ideal for this sort of application? So just the really simple basics of holography is that you have a beam of light, like a laser, that gets split into two beams. One beam serves as a reference, kind of what it looks like in that image when something's not there. And the other beam serves as the object beam. So for example, if we're looking at this tea kettle, supposedly a really tiny tea kettle under a holographic microscope, this beam tracks with the light, how it's altered by the tea kettle being there, and then we have the reference beam. So then we bring those together in a computer, and we can reconstruct the image and get this 3D image of what's going on in this microscope. So in bacteria, you can think of it as this tiny little cube of liquid that we can now see bacteria swimming around in 3D. So holograms in space might sound a lot like the movies, but I'm going to disappoint you a little bit. It doesn't look quite like that. So this is an example of some of the data that we get out of our microscope. So this is actually a hologram, although we're only looking at one single plane of the hologram. But what we're doing with this to capture motion is looking at video. So it might be a little hard to see with the lighting, but this, for example, is a little bacterium swimming through a field. And you can see there's tons of guys swimming through here. Does that look like a definitive biosignature? Would you say that's alive? Maybe. It can be a little hard to tell sometimes, but compared to some of the other stuff that's just hanging out in the field, for example, this little spot that's not really moving, it looks like that's alive, right? So it works pretty well in a laboratory setting. And then we can take it down to really low temperatures and see how bacteria might swim in these really cold environments like the Arctic or Europa. So up here on the top, we have these three different panels represent three different temperatures, so 6 degrees Celsius, -13, and -15, and we're putting these bacteria in these cold temperatures and seeing how they're swimming. So this is the hologram up here, and then we're looking at one plane of the hologram in each of these two images. So for 6 degrees, we take the hologram, we can track the bacteria using software and see their paths as they swim through the field. And you can see at +6, they're swimming pretty happily. There's lots of these colorful tracks in here. At -13 degrees Celsius or 8 degrees Fahrenheit, they start to slow down. The tracks aren't as plentiful. And at -15 degrees Celsius or 5 degrees Fahrenheit, it doesn't look like there's any bacteria swimming in there. So this data actually corroborates some other data that's been shown for this particular bacterium that we're using here in this data set, that -13 degrees Celsius is the known lower temperature limit of bacterial swimming on Earth. So this works in a lab, that's great, but as we all know, things in ideal environments don't necessarily always work well in the field like we might want them to. So we took this microscope up to the Arctic to see how it would do in this extreme field, to see if we could find life in this Arctic sea ice by looking at bacteria swimming. So we went up to Nuuk, Greenland, which is right here. So again, this is Alaska, and here's the North Pole. So we went up here to Nuuk, which in the wintertime has some pretty thick sea ice in the fjords that cut into the coastline there. So here's an example of what the terrain looked like out and around Nuuk. So this, all that we're standing on here, this flat is sea ice, so it's frozen seawater, presumably full of those bacteria and algae and those really salty brine pockets like we saw earlier. That's me. So we took this microscope out here, and of course we had to prepare the microscope for deployment in the field. So you saw this image earlier of the microscope, which is just this orange box, which looks kind of like there's not a lot going on. Here's our Shamu whale. But when you open it up, you can see that there's this assembly that looks kind of like a regular microscope you might see in a laboratory. So the laser comes through here, the sample gets put in here, and it goes through the different lenses and comes out into this computer. So we can just press the buttons on the side of the box after we've loaded into this sample chamber. So the sample goes into this little spot here, press the button, and then we can take the hologram, either take a still shot, a picture, or take a holographic video for a given amount of time. So that's what we did. We went out to Nuuk. So in that last picture, I was kind of lying to you. Nuuk is actually a pretty big town in Greenland. So you can see here some of the buildings. This is still sea ice out here, but we were also able to go out into further afield. This is us just sampling it on a smaller scale. So we take these ice cores here and drill some holes into the ice, set the microscope down inside the ice so it gets kind of that temperature like you would have in the ice. And then we take some liquid sample of those briny liquid pockets out of the ice and put them into the microscope and see if we can get nice holographic videos like we saw from the laboratory example. So the good news is we did get some awesome videos. So I'm going to show you a few of the videos that we got up in Greenland. So to kind of orient you, this is a still shot of what you're going to see in this larger video. So in this one, you're going to see a little bacteria, sorry, not a bacterium, a larger organism kind of swimming through the field. And we can tell that it's a larger guy and not a bacterium just based on the size, the scaling. So this is exciting. There hasn't really been images of things swimming in Arctic sea ice or any sea ice brines before. Which I guess on the whole scale might not seem that exciting, but for this application is pretty cool. So this is at -4 degrees Celsius or 25 degrees Fahrenheit. But what we're really looking for is bacteria that can swim in the sea ice. And we're looking for these especially small guys, the bacteria, because that's what we expect to find when we would go to Europa, for example. So here's a video of a really happy little guy just loving his trek through the brine. So you can see him swimming up through here. That's awesome. That's this really unambiguous sign of motion that we were hoping to see. And then we also saw some interactions between organisms in the sea ice. So here's one of those larger organisms like we saw earlier, and this little guy is kind of tracking around him. As you can see in this inset, kind of the whole image of his track. So these arrows are showing where he's swimming. The little guy, the little bacterium, and the larger organism just kind of lazily moves along. Okay, so that's cool. We can see things in Arctic sea ice, but what does that mean about life in outer space and ultimately the Shamu microscope? So like I mentioned, in addition to this sea ice covering on Europa, there's also this liquid ocean underneath. And earlier I said that everywhere we found water on Earth, we've found life. And so to give you an example of what that might look like on Earth, this is the Arctic Ocean. So this is a sample of ocean water from beneath the ice, and you can see how much more swimming there is in this example as compared to the ice. So if we can get into that ocean water, that's really a huge boon for the project. And what's especially unique about Europa is, in addition to this icy crust, it has geysers on the surface. And these geysers are coming from local hotspots inside the ocean and then thinner spots in this icy crust. And they're shooting through the crust a sample of liquid ocean. So for this image here, here's the crust. Remember, that's about 8 kilometers thick, or about twice as thick as the Antarctic ice cap. And here's the ocean underneath, and the geysers punch through softer spots in the ice and shoot out into space, which is really great from a mission standpoint if you're a NASA engineer, because now we don't have to worry about drilling through that ice. Oops, excuse me. We don't have to worry about drilling through the ice. The water is coming to us, right? If we can fly through that, take a sample of that plume, that's ocean water right there in our hands. So that makes it a lot easier for us to sample that ocean and hopefully find life. Now, Europa isn't the only place in our solar system where we might hope to be able to do this. So this graphic here is kind of akin to that GIF I showed you earlier, where this little ball up here represents the amount of liquid water on these different environments. So you can see Earth here has a decent amount. Mars has a lot of liquid water. You might remember that NASA press release in November about the recurring slope lineae as evidence of water on Mars. Here's Europa, so you can see it has about a comparable amount of liquid water to Earth. And then I also want to draw your attention to Enceladus, which is a moon of Saturn, not unlike Europa in that it has this at least partially global liquid ocean on it with an icy crust, and then also geysers shooting out of it for us to be able to sample. So there's lots of opportunities for us to hopefully use this microscope in outer space in addition to places on Earth. And hopefully the smallest bacteria, the smallest organisms on life will help us be able to find the answer to one of the biggest questions in humankind: are we alone in the solar system? So with that, I'd like to thank Town Hall and engage our funding sources, of course, UW Astrobiology and the School of Oceanography, and also our friends down at Caltech and NASA Jet Propulsion Lab. Thank you.
So I understand you're studying in the Arctic. What about Antarctica? Does it have briny veins that you expect, or have we already discovered those veins in Antarctica? Is that why you're now studying the Arctic Ocean?
Yeah, so the question was if there's also briny veins in Antarctic ice, and there is. So especially in sea ice, part of its formation is to get those briny pockets inside of the ice. So that happens both in the Arctic and sea ice and in the Antarctic and sea ice. What's different between the two poles is that Antarctica has a continent, so most of the ice on Antarctica is fresh ice. It's not sea ice, and it doesn't have as many of those salty pockets, so there's less life in that ice. So the Arctic is a really good system for us to study as an analog because it has the ocean underneath it that makes that saltier ice, and it's also a lot closer to Seattle. Any other questions?
Sure. So if you're unsuccessful in finding moving bacteria, what do you conclude?
Right. So if we don't find moving bacteria, that would of course be disappointing to the project. But one of the things about looking for signs of life in outer space is this concept that we need multiple converging lines of evidence. So even if we find swimming bacteria, just that one line of evidence isn't necessarily enough alone to say definitively that there is life. It's to us, obviously, it looks like there might be something swimming, but generally we like to see these multiple lines. For example, a chemical test that shows organics in the ice. This would be one line. So the idea is if we can get multiple lines of converging evidence, if we can see multiple different types of biosignatures pointing to life and environment, that's a really conclusive test. If we don't see multiple ones, we can make some inferences and hope that there might be life there, and then use that to kind of promote further investigations of Europa.
When is the mission to Europa, or has it already launched?
Yep, so when is the mission to Europa? It hasn't launched yet, so the idea is for the mission to launch in 2022, and then it takes 10 years to get there. So this is going to be really suspenseful for everybody. We're going to be waiting for a while. So there's a lot of planning and stuff that goes into these missions with these long projects, and a lot of waiting. So the team of engineers that works on this down at Caltech is really in it for the long haul. Sure.
Could you talk about how important sugars are to find in your experiment?
Absolutely, that's a great question. So she asked, how important are sugars in the experiment? So sugars are really important in icy environments because a lot of organisms that live in these icy environments produce tons and tons of sugars. It's called extracellular polysaccharide substances, or EPS, where saccharide is the sugar in that EPS. So these organisms make lots of EPS that coat them and acts as a cryoprotectant, protects it from some of that cold temperature that's in the ice. So if we can go to Europa and find sugars in the ice, that might be a really good biosignature that there's something there making this cryoprotectant, this EPS, that allows it to live in the ice. In addition to that, just finding sugars or general organic molecules is really encouraging for us that there might be life there if these organic molecules are being made by life. Mm-hmm. Great. Thank you so much for listening to the talk. I appreciate it.